Hi, everyone. My name is Hannah Safford, and I'm a third-year PhD student in Mike Mitchell's lab at the University of Pennsylvania. And today I'm excited to share with you all some of our work on ionizable lipid nanoparticles for in vivo mRNA delivery to the placenta. So the development of therapeutics for women's health applications is a severely underfunded and understudied field, and broadly, research in this space focuses on conditions that affect both non-pregnant patients, including reproductive tract disorders and vaginal infections, as well as conditions that affect pregnant patients, including placental disorders and congenital fetal disorders. Specifically, developing new therapies for pregnant patients is particularly challenging, as there are complex social constructs surrounding the inclusion of pregnant patients in clinical trials, and over 90% of clinically approved drugs do not have appropriate labeling information for pregnant patients. That being said, developing new therapies that can target the placenta is particularly important, as several different pregnancy complications can arise due to dysfunctional placental development. And for those of you that are less familiar with the placenta is an organ that develops during pregnancy, and we can use it to exploit targeted delivery of therapeutics either to the mother and the fetus. And broadly, the primary role of the placenta is to serve as this biological barrier between maternal and fetal circulation, protecting the fetus from any harmful molecules, as well as providing a site for nutrient-oxygen exchange to support fetal development. Just to quickly walk through some important kind of placental biology. During placental development, maternal spiral arteries extend from the uterine tissue through the placenta and supply blood to the chorionic villi, which is kind of the main site of nutrient and oxygen exchange in the placenta. And these chorionic villi are made up of a barrier of two layers of trophoblast cells, both the syncytiotrophoblastic layer and then a layer of cytotrophoblasts. And within these villi, there are fetal capillaries that transport oxygenated blood to the fetus to help support fetal development. And so as I mentioned, dysfunctional placental development can lead to several different pregnancy complications, one of which is preeclampsia, which affects about 5 to 8% of all pregnancies. And preeclampsia is characterized by the onset of hypertension after about 20 weeks of gestation, and can often be accompanied with other conditions such as kidney and liver damage, neurological conditions, and fetal growth restriction. And preeclampsia is thought to develop through a two-stage paradigm of placental dysfunction, where in the first stage we have improper remodeling of the uterine spiral arteries, which creates this low blood flow environment in the placenta and leads to oxidative stress and placental ischemia. This subsequently induces the second stage of preeclampsia, which largely involves the abnormal secretion of anti-angiogenic factors such as SFLT1 and the downregulation of vasodilatory growth factors such as VEGF and placental growth factor. And currently, there haven't been any therapeutics that have been developed to slow the progression of preeclampsia, and the only curative treatment option is the early delivery of the placenta in the fetus, which has its own set of complications. And so disease management largely focuses on symptom management for these patients, which involves the use of antihypertensive drugs to manage maternal blood pressure or the use of anticonvulsants to prevent seizures. And so we believe there's a significant need to develop new therapeutics that can mediate vasodilation in the placenta in order to help treat preeclampsia. And so in the Mitchell lab, we're broadly interested in developing new nanoparticle platforms that can deliver nucleic acids to a wide range of novel cells and tissue targets. And given this need to develop new therapeutics that can target the placenta, we're interested in developing new LNPs that can deliver nucleic acids to the placenta. And so, as we've discussed many times today, ionizable lipid nanoparticles have emerged as the most prominent non-viral platform for nucleic acid delivery, and broadly, LNPs can be used to correct dysregulated gene expression via transfection of host cells with exogenous nucleic acids. LNPs have demonstrated tremendous clinical success with the FDA approval of Alnylam's Onpattro siRNA therapeutic, as well as the emergency use authorization of the mRNA COVID-19 vaccines. And so LNPs are highly advantageous delivery vehicles as they can enable potent intracellular delivery and enable endosomal escape of their nucleic acid cargoes. And LNPs largely facilitate this endosomal escape due to the presence of the ionizable lipids in our LNP formulations, which are neutral at body pH, which reduces any risk of toxicity, but become charged at the endosomal pH, which helps disrupt the endosomal membrane and release our nucleic acid cargo into the cell. However, LNPs must overcome several of their own barriers to delivery in order to ensure the successful delivery of their cargoes. And namely, LNPs must overcome first-pass hepatic clearance and selective accumulation in their target organ of interest. These therapies must also avoid serum nucleases that can degrade their nucleic acid cargoes, as well as opsonization of their surface, which can lead to phagocytic clearance. And so given the many advantages of LNPs, we're interested in developing LNP therapeutics for placental disorders. However, since LNPs face many challenges with achieving extrahepatic mRNA delivery, we need to engineer new LNP platforms that can facilitate mRNA delivery to the placenta. To engineer these placenta-tropic LNPs, we can take an iterative approach where we can design new ionizable lipid structures to identify structures that can enable potent mRNA delivery to the placenta, as well as vary the relative molar ratios of the other lipid excipients in our LNP formulation, as it's been shown that the molar ratios at which these excipients are combined can be optimized for a variety of different applications, including encapsulating different nucleic acid cargoes or interacting with target cells of interest. And so today I'm going to kind of go over two papers from our lab. The first one, we synthesized a novel library of ionizable lipids to formulate LNPs and identified a lead ionizable lipid candidate that enabled selective extrahepatic mRNA delivery to the placenta compared to benchmark liver-tropic LNP formulations. Additionally, we also examined if there are any changes in the biodistribution of our LNPs in both non-pregnant and pregnant mice. In a follow-up study, we used orthogonal design of experiments to screen iterative libraries of LNPs with varied excipient molar ratios in order to identify a lead LNP formulation that mediated potent mRNA delivery to the placenta and mRNA transfection of placental trophoblasts, endothelial cells, and immune cells. So to begin, we synthesized our library of ionizable lipids using the fast and simple SN2 reaction scheme, where we reacted either one of 5 polyamine cores with one of 3 epoxide tails of varying length, either the C12, C14, or C16 tails. We then mixed each of these ionizable lipids with a phospholipid DOPE, cholesterol, and lipid PEG, and chaotically mixed these with mRNA to formulate our LNPs in a microfluidic device. And so to identify these placenta-tropic LNP formulations, we began by assessing in vitro mRNA transfection of our LNP library in placental trophoblasts, which, as I mentioned previously, is kind of the main cell type in the placenta. In particular, we choose to use a cell line JEG3 cells, as these are cells that have been previously used for in vitro models of placental trophoblasts. To screen our library, we encapsulated luciferase mRNA into our LNPs as a reporter cargo, where luminescent signal can be correlated to functional mRNA delivery and luciferase expression. And in addition to our LNP library, we treated our trophoblast cells with lipofectamine, which is just a commercial transfection reagent, as well as 2 industry standard ionizable lipids, either C12-200 lipid or the MC3 lipid. As you can see, after 24 hours of LNP treatment, we had 5 different LNPs that were able to mediate significantly higher luciferase expression compared to our lipofectamine control. And in particular, we selected 2 different LNPs to evaluate in vivo, namely the C12-4 formulation as our top performer, as well as the C14-5 formulation as a moderate performer. And then we also selected the C12-200 industry ionizable lipid as an industry comparison. And so we began by assessing mRNA delivery to the non-reproductive maternal organs, namely the heart, liver, lung, kidneys, and spleen, in both the non-pregnant and pregnant mice, to assess if we could identify any LNPs that can mediate extrahepatic mRNA delivery, as these same formulations may mediate mRNA delivery to the placenta. And in these experiments, we again formulated our 3 LNP formulations, either the C12, C14-5, or the C12-200 LNPs with luciferase mRNA, and administer them via tail vein injection to either non-pregnant mice or pregnant mice on gestational day E16, which corresponds roughly to the end of the 2nd trimester in humans. And as you can see in these images, the C12-200 LNP formulation mediated the highest luciferase expression in the liver, whereas the C12-4 LNP formulation mediated the highest luciferase expression in the spleen compared to the other two formulations. And we didn't observe any differences in luciferase expression between the non-pregnant and pregnant mice, but we did calculate a spleen-to-liver ratio for each of our treatment groups to identify if there are any changes in extrahepatic mRNA delivery between these different formulations. And as you can see in this graph here, for both the C12-4 and C14-5 formulations, they had higher spleen-to-liver ratios than the C12-200 particle in both the non-pregnant and pregnant mice, suggesting that these formulations are capable of achieving extrahepatic mRNA delivery. We also noticed that there is a higher spleen-to-liver ratio in the non-pregnant mice when compared to the pregnant mice for the C12-4 and C14-5 formulations. And we think this may be due to the fact that during pregnancy, we're seeing partitioning of this extrahepatic mRNA delivery between both the spleen and the liver. And so if we examine luciferase expression in the placentas, you can see here that the C12-4 LNP formulation was able to mediate significantly higher luciferase expression in the placentas compared to both the C14-5 formulation and the C12-200 formulation. Additionally, we didn't observe any signal luciferase expression in the fetuses, and we think this is due to the size of our nanoparticles, as they're around roughly 100 nanometers, which should prevent any placental transport and entry of these LNPs into fetal circulation. And so having successfully identified a lead ionizable lipid for potent in vivo mRNA delivery to the placenta, we are interested in further optimizing our LNP formulation for enhancing mRNA delivery to the placenta. And so in this study, we were interested in optimizing the molar ratios of our excipients in our LNP formulation, namely the ionizable lipid, cholesterol, the phospholipid DOPE, and lipid PEG, as the excipient molar ratios that people typically use and what was just used in all the data I've just shown you were ratios that were previously optimized for mRNA delivery to the liver. And one of the main techniques that's used to optimize excipient molar ratios is orthogonal design of experiments. And this technique allows us to design large libraries of LNPs and screen these formulations based off of a few representative formulations to identify optimized molar ratios for mRNA delivery to a target organ of interest. And so in our first library of LNPs, we used the top-performing ionizable lipid that we had previously identified, the C12-4 lipid, as well as DOPE, cholesterol, and PEG, and examined them at 4 different molar ratios to create this representative library of 16 formulations. And after LNP treatment, we again used luciferase mRNA for these experiments to quantify mRNA transfection in trophoblast cells. And so after LNP treatment, we compared luciferase expression across our library to this LNP formulation, LNP-S1, which is the formulation made with the standard excipient molar ratios that we've used previously. And as you can see in this first library, we were unable to identify any LNP formulations that were able to significantly improve mRNA delivery to trophoblasts compared to the standard formulation, with the exception of LNP-A1, which had comparable delivery. And so while none of our formulations were able to enhance mRNA delivery over the S1 formulation, the relationship between the different excipient molar ratios and mRNA delivery can be investigated to identify trends that lead to relative improvement in mRNA delivery to trophoblasts. And so to do this, we can plot the average luminescence signal of the 4 LNPs that have the same excipient molar ratio. And I won't go through all the excipients here, but if we focus on both the ionizable lipid and the phospholipid, we can see at both low and high molar ratios of our ionizable lipid lead to improvements in mRNA delivery to trophoblasts, whereas for our phospholipid DOPE, we're only seeing improvements at lower molar ratios. And to further inform our library design, we can investigate the impact of excipient interactions on trends in mRNA delivery. And to do this, we can plot the average luminescence signal of LNPs with the same molar ratio of one excipient with either the lower or higher molar ratios of a second excipient. If we focus just on the ionizable lipid here for these graphs, we can see that at low and high molar ratios of our ionizable lipid, we have improvements in mRNA delivery with low molar ratios of DOPE. Whereas if we look at the relationship between our ionizable lipid and cholesterol, we have improved mRNA delivery with low molar ratios of our ionizable lipid and cholesterol, whereas we have improved mRNA delivery at high molar ratios of our ionizable lipid with high molar ratios of cholesterol. And so using these trends, we designed two sequential 8 LNP libraries, again using orthogonal design of experiments within a narrowed range of excipient interactions, where library B was optimized around low molar ratios of our ionizable lipid, and library C was optimized around high molar ratios. And as you can see in this graph, in library B, we are able to identify one LNP formulation that significantly improved mRNA delivery over our standard formulation, whereas in library C, almost all of the formulations demonstrated improvement in mRNA delivery to trophoblasts. In particular, LNP-C5 was able to mediate a 4-fold improvement in mRNA delivery compared to LNP-S1. And to confirm the results of our initial library screens, we selected the top performing LNPs from each library, namely LNP A1, B5, and C5, along with S1, and evaluated them in a dose-dependent response. And as you can see, across all these doses tested, LNP-C5 was able to mediate improvements in mRNA delivery compared to LNP-S1. In addition, we noticed that LNP-A1 was able to mediate the greatest improvement in mRNA delivery compared to S1 at the highest dose tested, and so we selected both of these formulations to further evaluate in vivo. And so again, to evaluate in vivo mRNA delivery to the placenta for our optimized LNP formulations, we again administered LNPs A1, C5, and S1 encapsulating luciferase mRNA via tail vein injection to pregnant mice. And if we first look at the maternal organs, we can see that across all of our LNP formulations, we have the highest luminescent signal again in the livers and the spleens. And in particular, LNP-C5, we saw a reduced mRNA expression in the livers compared to the other two formulations, and LNP-C5 was also able to mediate enhanced mRNA delivery to the spleen, suggesting that it can mediate potent extrahepatic mRNA delivery. And so when we look at the placentas and the fetuses, we can see that for LNP-C5, it's able to significantly improve mRNA delivery compared to both the standard formulation as well as LNP-A1. And again, we don't see any signal in our fetuses suggesting that these LNPs remain in the placenta, kind of highlighting the safety profile of this LNP platform to solely remain in maternal circulation. And so next, we are interested in evaluating mRNA delivery in the different cell types of the placenta of our optimized LNP formulation. And I previously mentioned trophoblasts and their role in the placenta microenvironment, but we're also interested in evaluating mRNA delivery to immune cells and endothelial cells, as all three of these cell types play important roles in the pathophysiology of placental disorders and are able to secrete growth factors into the placental microenvironment. And so for these experiments, we encapsulated mCherry mRNA into LNP-C5 and S1 and administered these LNPs via tail vein injection to pregnant mice. We then isolated the placentas and stained them for flow cytometry in order to evaluate mCherry expression in these three different cell types. And here we show that LNP-C5 is able to mediate mRNA delivery to placental trophoblasts, endothelial cells, and immune cells, and that demonstrates moderate improvement in mRNA delivery over the S1 formulation in all three of these cell types. And so these results suggest the ability of our LNPs to deliver mRNA to three key cell types in the placenta, which are all important for treating placental insufficiency disorders. And so having only used reporter mRNA cargo throughout this screening process to engineer our placenta-tropic LNP formulations, we wanted to evaluate the functional delivery of a clinically relevant mRNA cargo for placental disorders. And so in these proof of concept studies, we encapsulated vascular endothelial growth factor, or VEGF, into our C12-4 LNP formulation. And we chose to evaluate VEGF here for these studies as it has reduced bioavailability during preeclampsia and plays a role in remodeling the placental vasculature. Additionally, there have been some previous studies that have evaluated recombinant VEGF protein therapies and VEGF adenovirus-mediated gene therapies for placental disorders, including preeclampsia. And so for these studies, we used healthy pregnant mice and treated them with either PBS or the C12-4, C12-200 LNP formulations encapsulating VEGF mRNA. And here we evaluated the serum levels of VEGF at both 6 and 48 hours after LNP treatment. And as you can see, at 6 hours, VEGF levels were significantly elevated over the PBS controls for both our C12-4 and C12-200 LNPs. However, these levels returned to baseline after 48 hours. And we think the high levels of VEGF secreted by the C12-200 LNP formulation is due to its ability to potently transfect the liver and the efficacy of protein production by liver hepatocytes. We then assessed LNP-mediated inflammation in the placenta at both 6 and 48 hours after LNP treatment, as we want to minimize any LNP-associated inflammation in the placenta, since during preeclampsia there's already pre-existing inflammation and we don't want to exacerbate this further. And so here we selected 7 different cytokines that have been shown to mediate inflammation in the placenta and assessed their relative concentrations of each cytokine in our LNP-treated mice compared to the PBS controls. And as you can see, this is something that's typically observed at acute time points after LNP treatment. The relative cytokine concentrations were elevated for both the C12-4 and C12-200 mice, but these levels returned back to baseline by 48 hours, suggesting that there aren't any long-term inflammatory effects of our LNPs. So we also assessed LNP-mediated liver toxicity by measuring the serum levels of either the secreted enzymes, either ALT or AST, as these enzymes are often used to assess nanoparticle-mediated toxicity, as elevated levels of these enzymes can indicate hepatic injury as a result to potent nanoparticle accumulation in the liver. And as you can see, we didn't see any differences in the ALT levels across our different treatment groups, but we did see elevated AST levels mediated by our C12-200 LNP treatment, suggesting that there may be some toxicity as a result of our C12-200 formulation. And so to supplement our VEGF data, we also wanted to utilize immunohistochemical staining to evaluate the upregulation of the VEGF receptor 1 in response to an increase in local VEGF expression. And in these photos, the positive VEGF-R regions are stained brown. And as you can see in these images, there's an increase in the brown stage regions, particularly for our C12-4 LNP treated group. And if we quantify the positive areas with ImageJ, we can see that we have an increase in the positive VEGF-R1 areas for both our C12-4 and C12-200 treated mice. In addition to assessing local VEGF expression, we wanted to evaluate functional VEGF-mediated vasodilation in the placenta, and we did this using H&E staining. And in these images, the white areas are representative of both maternal and fetal blood vessels, while the dark purple, large brown regions are our trophoblasts, and the smaller purple stained nuclei are our endothelial cells. And compared to the PBS-treated mice, we see an increase in the blood vessel area for both our C12-4 and C12-200 LNP formulations, and we think we have slightly more homogeneous blood vessel area for our C12-4 treated mice compared to the C12-200 treated mice, which we think may be a result of local versus systemic VEGF secretion. So to conclude, we were able to successfully design and engineer a library of ionizable lipids for mRNA delivery to the placenta, and we were able to utilize orthogonal design of experiments to further optimize our LNP formulation for in vivo mRNA delivery to the placenta and potent mRNA transfection of placental trophoblasts and endothelial cells. We were also able to demonstrate a proof of concept study for in vivo VEGF mRNA delivery to the placenta in order to mediate placental vasodilation during preeclampsia. And lastly, future directions to clinically translate this work will involve evaluating our placenta-tropic LNP formulations in a murine model of placental insufficiency. With that, I'd like to thank all my members of the Mitchell Lab, in particular my PI, Dr. Mike Mitchell, and Kelsey Swingle, who's another PhD student in the lab, who also helped spearhead a lot of this work. And with that, I'm happy to take any questions.
Such a great presentation. Thank you so much. So any questions? There's one up there.
Yeah, I think that's definitely something we're concerned about. And so, interestingly, guinea pigs have pretty similar placental biology to humans. And so I think kind of the next or future step of translating this work is moving into larger animal models to kind of see if would be close to what we observe in humans. Yeah. Yeah. Yeah, let's see. I believe the pKa of the formulations we're using are around 6.5, which I think is pretty standard for a lot of the different ionizable lipids. And I think it's definitely important, I think, in staying in that lower pKa region in order to ensure that we do have kind of the switch and the ionizable lipid becoming charged at that endosomal pH. So yeah, that's definitely something that we need to consider moving forward. Yeah.